For over a century, the study of oxytocin has focused on reproductive physiology and social behavior, specifically the mechanics of childbirth, lactation, and maternal-infant bonding. Geroscience is now examining the peptide through a different lens. In a study by Majima and colleagues, researchers administered a daily intranasal dose of oxytocin to biologically aged mice for only 10 days. This graph illustrates the response during those 10 days. As intranasal oxytocin enters the system, we see a sharp rise in CFOS, a marker of neuronal activation, paired with a simultaneous drop in the inflammatory marker hsCRP. The observation becomes more significant past day 10. Once the external treatment stops, the animal's endogenous plasma oxytocin levels do not return to their previous aged state. Instead, they remain steady at a youthful, elevated level. This suggests the 10-day intervention did more than just supplement a deficiency. It appeared to trigger a self-sustaining internal production that persisted long after the final dose. Understanding how a brief exposure leads to a lasting biological change requires looking at the transcriptional environment of the aging mammalian brain. The decline of the neuroendocrine system isn't typically the result of irreversible neuronal death. In the aging hypothalamus, the neurons are largely intact, but they have become functionally dormant. This dormancy is driven by epigenetic drift, a process where gene expression is progressively silenced by chemical modifications to the DNA. The evidence suggests that oxytocin functions as an epigenetic regulator, capable of reversing this silencing and restoring the transcriptional activity of these dormant neurons. To identify how the system restarts itself, we must deconstruct the accelerating feedback loops that cause a mammal to biologically degrade in the first place. The process originates in the oxytocin-producing neurons of the paraventricular and supraoptic nuclei within the hypothalamus. This flowchart maps the sequence of decline. The first stage is the hypermethylation of the oxytocin gene promoter, where methyl groups physically block the cell's ability to read the DNA. This silencing acts as a second domino, causing a severe downregulation of the enzyme TET2. TET2 is the very machinery the cell needs to remove those methyl marks and keep the gene active. Without TET2, the neuron's mitochondria begin to fail. Specifically, the expression of respiratory complex 4 collapses, plunging the cell into an energy deficit while increasing the leakage of reactive oxygen species. This local oxidative stress invites a systemic response. Inflammatory cytokines like TNF-alpha infiltrate the brain, further suppressing the remaining oxytocin production. These cytokines reinforce the initial gene silencing, closing a loop that accelerates the aging of the entire neuroendocrine network. Once this cycle is closed, the biological decay of the hypothalamus becomes self-perpetuating, requiring a specific molecular signal to physically break the loop. The intranasal delivery of oxytocin provides that signal by moving directly into the central circulation, bypassing the blood-brain barrier. Once in the hypothalamus, the peptide targets the specific neuronal clusters that have been silenced by the aging process. The success of this intervention is dependent on the oxytocin receptor, or OXTR, which must be present to process the incoming signal. When the oxytocin molecule docks into its receptor, it triggers an intracellular cascade, carrying the signal from the cell membrane toward the dormant nucleus. This signal is specifically calibrated to reach the hypermethylated chromatin that has kept the oxytocin genes silenced for years. Without this continuous receptor signaling, the molecular machinery required to reverse epigenetic aging remains locked and inactive. Inside the nucleus, the DNA is tightly coiled into structures called nucleosomes, which are covered in the repressive methyl marks associated with advanced age. The signal from the oxytocin receptor recruits the TET2 enzyme, directing it to bind to the silenced transcription start sites. TET2 then pairs with OGT, a metabolic enzyme that acts as a nutrient sensor within the cell. These two molecules form a synergistic complex on the genome, preparing to physically alter the structure of the DNA. OGT begins the process by attaching a sugar molecule to a histone, the spool around which the DNA is wrapped. This specific post-translational modification is known as O-GlcNAc. The modification causes the nucleosome to loosen, and the tightly wound DNA physically unravels, exposing the genetic sequence for the first time in decades. By forcing the chromatin into this open state, the cell transitions the genome from an inaccessible, dormant architecture into a youthful, active one. Once the DNA is accessible, the complex begins the final stages of epigenetic remodeling. The TET2 OGT team recruits a third partner, the SET1/COMPASS complex. This new complex applies a trimethylation tag directly to the tail of the histone protein. This specific marker is designated as H3K4ME3. In the language of the genome, this tag is the definitive signal for the cell to begin active transcription. Finally, TET2 moves linearly down the DNA strand, acting as an epigenetic plow. It oxidizes the repressive 5-methylcytosine marks, physically stripping the age-associated methylation from the DNA sequence. This coordinated strike erases the effects of epigenetic drift, allowing the dormant gene to resume its youthful function. As the oxytocin gene is unsilenced, the hypothalamic neurons immediately resume the production of oxytocin mRNA. This internal production restores mitochondrial health. The cell begins synthesizing respiratory complex 4 again, resolving the energy deficit and halting oxidative stress. The restored oxytocin signaling also actively suppresses the HPA axis, shutting down the systemic production of inflammatory markers like hsCRP. In our flowchart, the cycle now reverses. The downward spiral of aging is replaced by a self-sustaining virtuous circle of molecular maintenance. Because the neurons have regained their innate capacity to produce the peptide, the system can maintain its own health without further external intervention. Restoring the hypothalamic oxytocin system creates a cascade of biological effects across the entire body. In peripheral tissues, oxytocin targets muscle satellite cells. This restores their regenerative capacity to youthful levels and begins reversing the muscle loss associated with aging. In the skeletal system, oxytocin shifts stem cell differentiation toward bone-forming osteoblasts, helping to preserve bone mass and slow the progression of osteoporosis. It also provides cardiometabolic protection by enhancing glucose uptake and dampening the inflammation that drives the buildup of arterial plaque. By reversing epigenetic drift driving neuroendocrine failure, oxytocin acts on the genomic causes of decay. This research identifies it as a fundamental regulator of the aging process itself, offering a scientifically validated path to preserving molecular fidelity across the lifespan.